Good morning, Dr. Meneses.
Good morning. O pa sila mahuman?
Mana sila, Doc. Then Nilwen is not yet here.
Oo, wala ka nila.
You'd like to go ahead, Doc? Or you have to wait for him?
Saan ba? Pwede na kailangan ng delivery. Oo! Matungod ang langas. So, I'm going to expand. Okay. So, good morning everyone. Aha, kaya ko pakita sa akong slideshow. So, this is, I will go ahead na.
Yes, Doc.
Okay. As you will see in the first slide, you have the chromosome, then from there you have the different genes, and then each gene is made up of double helical DNA structure wherein you have, so these are the part of the DNA, excuse me. These are the part of the DNA element that is expressed, and then the intervening sequence, this does not code for any RNA or DNA or for protein. And you have also different kind of regulatory sequence. So then you have transcription, and then there is the post-transcriptional modification. And then from the different RNA, you can now will be able to translate it into a particular protein. So the ultimate expression of the genes in the chromosome is to produce protein for the cells. Okay. So we will end until the genetic code. So this is the central dogma of molecular biology. First, DNA replicating itself, exact copy of itself is replication. Then from DNA, you can transcribe to the different RNAs. Then from RNA, you need your mRNA, tRNA, and the ribosomal RNA to translate it into protein. Okay, you have arrow from RNA to DNA, so that is reverse transcription, or reverse transcriptase is the enzyme. So DNA to RNA transcription, RNA to polypeptide chain, so translation. Then the folding will have to happen, then it will become the functional protein as the tridimensional structure or the tertiary structure as well as the quaternary structure for protein having more than one polypeptide chain. So the genetic information stored in chromosomes and transmitted to daughter cells through DNA replication is expressed through its transcription to RNA and subsequently translated into protein. So all cells undergo a division cycle during their lifespan. During the process of cell division, everything within the cell must be duplicated. This is so that it can ensure the survival of the two resulting daughter cells containing now each of their DNA. Of particular importance for cell survival is the accurate, efficient, and rapid duplication of the cellular genome by way of the DNA replication. So the function of the DNA replication is to provide progeny with the genetic information of the parent cell. It occurs with high fidelity to maintain the genetic stability within the organism as well as the species to which the organism belongs to. Then the manner of replication is the so-called semi-conservative process or manner of replication. The replication is a complex, highly coordinated series of reactions, and both of the DNA strands serve as the template, and it is multifocal, so it can occur in many points along the DNA strand, and it is bidirectional. Okay. Now the direction of replication, transcription, and translation is from 5' to 3' direction. So we have what we call the continuous or the leading strand that follow the opening of the replication fork, and the one that is replicated away from the replication fork is the discontinuous strand or the lagging strand. It will require a primer, which is a short RNA stretch about 10 nucleotide long. So RNA primer is a prerequisite for DNA replication. As you will notice in the semi-conservative manner, each of the parental strand, the one in blue, is the parental strand. They serve as template, and then a new strand will be formed. So in essence, the daughter DNA is made up of an old strand from the parents and a new strand. And that is the meaning of semi-conservative. Okay. What we are showing here is, first we have the so-called corre element on the top, and you have the X. Okay. Then before that, there is an AT rich region. So the AT rich region differ from the GC rich region in that it has 2 hydrogen bonding between the AT and 3 hydrogen bonding between the GC. So it is easier to unwind with the AT rich region. Okay. Then the one that will recognize the ORI element is obviously your ORI binding protein. Okay. Then there will be formation of the replication fork for initiation of replication down the line. Okay, so I have another illustration. So on one side is the function of the protein or enzymes involved in replication. On the other side is the name of the protein or enzymes. Okay. Prevent premature reannealing of double-stranded DNA, meaning to say the double-stranded DNA has to open. Okay, so it will now expose the template on the DNA strand. So to prevent them from joining again, you need your what? SSB protein, which stands for single strand binding protein. Okay. Then you have processive unwinding. This is acted upon by the helicase. It will need an ATP to separate the two strands. Then it will initiate the synthesis of the RNA primer by your primase. Okay. Then it will relieve the torsional strain. What does this mean? It means to say, you imagine a double-stranded rope. Then as you stretch it at one end, the one behind the opening will be tightened. Okay, so it has to be relieved. This is the action of your topoisomerase, which actually is a bifunctional enzyme in the sense that it can cut or it can nick and then it will join. Okay, this is your topoisomerase. Then deoxynucleotide polymerization is the action of your DNA polymerases. Then the sealing of the DNA nick between the nascent chain and the Okazaki fragments. These are the fragments found in the discontinuous strand or the lagging strand. So this will be joined by your DNA ligase again. This will require your energy ATP. So the unwinding and the joining will need ATP. Okay. Why is there a need to have the RNA primer? The reason for that is to provide the so-called free 3' OH group. Okay, so it will now enable the DNA polymerase to look at the last bullet. So there will be a nucleophilic attack by the 3' OH group to the alpha phosphate of the incoming NTP with the formation of a phosphodiester bond and elimination of the pyrophosphate. Okay. So after that, depending now on what is the incoming, it will now dictate as to what will be the next nucleotide. Okay. So this is the illustration. As you will notice, the base, the 80 or the GC can have hydrogen bonding to stabilize, okay, the formation of the new strand. Okay, so you have your free OH group. It can now do a nucleophilic attack on the alpha phosphate group of the incoming nucleoside triphosphate. And then there will be the release, the red one, the bond that is broken is your pyrophosphate group. So the formation of the bond is 3' to 5' phosphodiester bond. Okay. So it is from 5' to 3' direction. The ability of the polymerase to remain associated to the template is called processivity. The longer it associates, the higher the processivity of the enzyme DNA polymerase. And this is enhanced by additional of protein activity of the replisome identified as processivity accessory protein. You will notice that the molecular processes of replication, transcription, and translation requires a lot of accessory protein. It will just carry the name of what it is supposed to help in the process. Okay, these are the differentiation between the prokaryotic, the bacterial DNA polymerases, and the eukaryotic DNA polymerases. Hmm. For the bacteria, we have the DNA polymerase 3, then DNA polymerase 1 or 2, then DNA polymerase 1. As you will notice, you have your 5' to 3' polymerases because that is the direction of the replication. Then you have proofreading, the 3' to 5' exon nuclease. The proofreading is telling us that if there is a wrong base that has been added, it can actually be recognized by your DNA polymerase, and it will remove that and it will put in the correct base. Okay. And then what is the role now of 5', 3' exonuclease? This is the one that will clip your RNA primer. And it will be replaced now by your deoxyribonucleotide. Okay, remember you have the primer. It cannot exist in the daughter DNA molecule as having an RNA element in the DNA. Then in the eukaryotic on the other side, uh, how can I remember the alpha, beta, delta, epsilon, gamma? Alpha, okay, the first thing that will be formed is the RNA primer, so that's why it's the alpha. Then beta is the specific DNA polymerase that is needed to repair the DNA. Okay. Okay. Delta and epsilon. The epsilon looks like the letter E, correct? I am telling you that you should know how to appreciate what you are looking at to help you remember. So the epsilon is like the letter E in the leading strand. And then the delta is like an inverted G in the lagging strand. Are you looking at it? And then we have also DNA in the mitochondria. So the gamma DNA polymerase will replicate the mitochondrial DNA. Okay, so alpha, beta, delta, epsilon, gamma. So the delta, epsilon, and alpha is involved in DNA replication, the beta for repair, and the gamma for mitochondrial DNA replication. This is another illustration to help you see. Okay, you have your RNA primer, then it will join by the DNA ligase. Okay, then you have your delta, then you have the epsilon, the one that is moving following the opening of the replication port is the leading strand. The one that is formed away from the replication port is the lagging strand. Then you have the helicase unwinding the double-stranded DNA, then the single strand binding protein to stabilize the single strand, and then the topoisomerase to relieve the torsional string, which is bifunctional enzymes. Okay. So you just listen to this one. The cell cycle is defined as the sequence of events that occurs during the lifetime of a cell. It is divided into 4 major periods. Okay, so all cells undergo a division cycle during their lifespan. So some cells continue to divide, exemplified by your stem cells. Okay, while the other cells divide a specific number of time until the cell death occurs, and this is the normal cell death, which is referred to as apoptosis. Then most cells will divide a few times before entering into a terminally differentiated. So among the cells in the body, for those who form tissues, actually it started as undifferentiated and then it will move up to the differentiated type. And this will actually tell you that the differentiated is the more mature cell rather than the undifferentiated. And that is the reason why in malignancy, it's kind of a worry if the malignancy is of the undifferentiated type because it can transform into something else, okay? Or it may go into a quiescent state. So the four phases or period is the M, G1, S, and G2. M is the period in preparation for mitosis to happen. The G1 is, this is that period of the cell cycle wherein it will decide, will I continue to divide or I will go into the quiescent state. If the decision is to go to the quiescent state or it has terminally differentiated already, then it will go to the GO phase. Okay. Now in the GO phase, the cell actually in a state of extended periods of time. Stimuli can actually induce the GO to re-enter into the cell cycle and then the G1 phase, and then it will induce a permanent terminal differentiation. Okay. So G1, the S, preparation, synthetic replication, synthesis, then G2 phase. So typically the eukaryotic cell membrane can be as short as 6 to 8 hours or as long as 100 days. The high variability of the cell cycle times is due to the variability of the G1 phase. The G1 phase is the phase prior to the replication process. So in the G1 phase, all the necessary cellular components, the different enzymes, the different protein that should be there will be formed, will be available. Okay. This is from Histo book. Just to show you that the cell cycle of the cell is well regulated. So there is an internal quality control mechanism at each point. Okay. Then, so these are the example of the damage that will happen in a particular phase of the cell cycle. This one you have to remember. This is important. Okay. So during the S phase, one and only one replication happen. And the marker to tell us that replication has occurred already is the methylation of cytosine. Okay, if there is a methylation of the cytosine in the DNA, then it means to say replication has happened. In addition to that, we actually have proteins that are involved. Proteins known as cyclins, okay, because this is a cell cycle activity. Cyclin, which can be turned on at the appropriate time, and they are called your cyclin dependent kinases. So the cyclin will be phosphorylated by the cyclin-dependent kinase CDK to phosphorylate the substrate, which is essential for progression through the cell cycle. And we have this combination of the cyclin and the CDK that is involved in the G1 phase for it to pass through the restriction point, the arrow, okay. And it says here, it will phosphorylate the RB. Okay. And it will allow it to pass through the so-called restriction point. And then you need to set cyclin A acted by CDK2 and cyclin E acted by CDK2. And then the last one, cyclin B and CDK1. Okay. This is just to show you that RB is actually retinoblastoma protein. The RB is a cell cycle regulator. So E2F is a transcription factor. So it will act on the histones and the DNA replication protein. So what happened originally is the RB is attached to your E2F, okay? So in the presence now of your cyclin D and CDK4/6, so this is really complicated, meaning to say the activation would require obviously the growth factor passing through the RAS pathway, and you need ATP. So what will happen? It will phosphorylate the RB protein and allowing the transcription factor to transcribe the DNA. Okay, and then RNA translation. So you will now have the different enzymes and protein that is needed for the replication to happen in the next phase, which is the S phase. So this is at the late G1 phase, passing through the restriction point, so that replication can happen. Okay, that is the end of replication. We will now go to transcription. So DNA gene can be transcribed to RNA. This is highly selective biochemical differentiation of the different tissues. All mammalian cells contain the same genome. However, it depends on what in the information in the gene that will be transcribed to become protein that will differentiate the renal cell from the muscle cell, so that a particular cell can perform a specific function. Now, the difference with the replication is that the primary transcript or the newly formed product of transcription, known as the primary transcript, is not active. It needs to be modified to become the functional RNA. Okay? There is no primer, there is no proofreading. The proofreading of the replication is to guarantee the fidelity and accuracy of the replication process. So we have the template strand, that part of the DNA that serves as the template for transcription, and then the opposite strand is known as the non-code. The opposite strand is known as the coding strand, because if what is transcribed is the mRNA, then this can become the template for translation. So the transcription units start with the promoter site, ending at the terminator element, the promoter element. Read out 3' to 5'. Again, the process proceeds from 5' to 3'. As pointed out here, gene A is the one of the strand, and then for gene C, it is the opposite strand. So any of the strand in the DNA that can be expressed is served as your template for transcription. So the promoter is upstream, you need to say before. Downstream is after. So the promoter is upstream, Then you have the RNA coding region until it reaches the terminator element. Okay, we will talk about the prokaryotic transcription. It is a very large protein or enzyme, a DNA-dependent RNA polymerase. Obviously, the template is the DNA. Then RNA holoenzyme, it is a metalloenzyme. It contains zinc. The RNA holoenzyme is made up of the core complex and the sigma factor. Okay, the sigma factor is very important here. Now the core complex is 2 alpha, BB prime, and omega. So we have the template binding, then it will initiate, then we have what we call the promoter clearance, elongation, and termination. So the molecular process starts with initiation, elongation, and termination. So one RNAP may be able to transcribe the same template by phasing it and spacing them. Okay. So the first is template binding. What happened here is that the RNAP holoenzyme binds a DNA part. Okay. Then it will scan. As it scans, it will recognize and bind to the promoter site with high fidelity. So the one that is able to recognize and bind to the promoter site is the function of the sigma factor. So the sigma factor recognizes the promoter site and allow the RNA polymerase to bind with high fidelity so that it can now start the transcription in the right position. That is the reason why the promoter element or the promoter site is before the initiation point of transcription. Okay. So first the promoter is closed, then it will be open. Okay. So initiation will happen. So the DNA template is read from 3 to 5 prime and synthesize RNA from 5 prime to 3 prime. Now this is an always thing. The first nucleotide that is laid down is either an A or a G at the 5 prime end, and it is your triphosphate. So an ATP or a GTP is the first transcribed nucleotide of the or transcription. So that will serve as the initiation site, and then polymerization will happen. When the ribonucleotide that is formed is about 10 to 20 ribonucleotide long, the sigma factor is released. So there is now promoter clearance. So it will continue to the elongation phase. So the elongation complex, so this has a lot of function in itself. It can continue to unwind the template to gain access to the template. And then after it has done it, it has to rewind the DNA. And then it has also to have the topoisomerase. So the bubble, transcription bubble, consists of about 20 base pair per polymerase. So first is you have the closed complex, the open complex, the initiation, then clearance, then elongation, and termination will happen when it reaches the terminator element that is described as hyphenated or having inverted interrupted repeats of 80 base pair. So it actually look like an intramolecular hairpin structure on the transcript. It will require another factor, the Ru protein, which is the terminator protein that will need your ATP RNA stimulated helicase. So this is the one that will recognize the termination of the terminator element. And this is how the inverted hairpin-like structure looks like. So it can bring about the ending of transcription. This is something new in the 31st edition. I will not touch on this. This is the part for Dr. Salses. And also what is written about is the function of the other. And what I want you to remember is this one for the RNA polymerases found in the eukaryotic. So there are 3 types. And as you will notice, the first transcript that is formed is the pre-RNA. Okay. The 18, 5.8, and the 28 srRNA. These 3 are what we call the 45 srRNA. So that is the precursor. Okay. So 5.8, 18S, and 28S RNA is the precursor, or the pre-rRNA is referred to as 45S RNA. So it is catalyzed by RNA polymerase 1. RNA polymerase 2 will form your pre-mRNA, or the other name is HNRNA. And then it matures to become the mRNA. Okay. Then the last RNA polymerase is your pre-tRNA, the 5S, okay, RNA. And the enzymes, the RNA polymerases, are differentiated from one another in how they are affected by your alpha amanitin. This is a Mostrom principle. So the 1 is not sensitive, the 2 is intermediate, and the 3 is sensitive. Sorry, the 2 is high and the 3rd is intermediate, basing on the microgram per ml. Okay. For translation to happen, we need the mRNA, the 1, 2, 3, 4 rRNA, and the tRNA. Okay. Now, each type of RNA polymerase is a complex of many polypeptide subunits, as many as 14, and it is 500 to 700 kilodalton. Each will require binding of transcription factors to locate the promoter and orient the polymerase correctly. Okay. Now, what you are seeing here is the regulated expression and the basal expression. Okay. The regulated is what we call the regulatory elements. If the effect is positive, then they are called the enhancer. If it is negative, then it is called the repressor. As you will notice, the regulatory element is found either upstream or downstream. Okay. Now, what is the meaning of data box and the GC or CAAT box? which is part of your promoter. The tata is part of your promoter element. For the eukaryotic, it is what we call the initiator element or the DPE downstream element. The tata box will actually what? Dictate where to start the transcription. Okay, well, the CAAT box, this is prokaryotic. The GC box is for eukaryotic. It will dictate how many transcript will be formed. So one is where, the other one is how many. A hormone response element can either be enhancer or repressor. So the eukaryotic transcription polymerase complex is truly a complex apparatus, very large. It has 50 unique proteins. It will provide for accurate and regulatable transcription. It has a lot of transcription factor. The basal components, you have to know what they are, but you have to understand that it will need activator as well as co-activator. Okay? So TBP is transcription factor binding protein. The 2 there stands for polymerase, RNA polymerase 2. So this is the one that will be transcribing to form your HN RNA, then post-transcriptional modification to become the mature mRNA. So first, it is very large. It requires a lot of transcription factor, co-activator, activator. And then, what is this telling us? So, to make everything simple, the transcription factor will bind to the enhancer DNA, okay, upstream of the promoter. And it will recruit chromatin remodeling and modifying co-regulatory factors. Never mind what they are. So the enhancer will, at this time, it is upstream of the promoter and it can recruit chromatin remodeling. What is being remodeled in the chromatin? Unfortunately, you have your group discussion already of what is in a chromatin. So it actually needs to evict the nucleosome. Okay, it needs to evict the nucleosome so that the promoter will be open or is now open, ready for transcription. Okay, these are all the same thing. ATP obviously is needed to remove the nucleosome. Now, there are 2 models for the assembly of the pre-initiation complex. So for the eukaryotic, it is very important that there is the formation first of the pre-initiation complex. But before that, the nucleosome has to be removed by many of the other protein factors involved. And then the assembly of the complex, pre-initiation complex, is one after the other, or by recruitment and then putting it there. We will look at the stepwise assembly of the pre-initiation complex. First, you have to understand that phosphorylation of the enzyme is important at the CN of the protein enzyme. Okay, so it has the heptap, the so-called repeat domain where the kinase can act on to put a phosphate group to make the eukaryotic RNA polymerase 2 active. Okay. Now, the G is made up of the binding transcription factor binding protein and 14 accessory factors, transcription factor, accessory factors. So the D is the one that binds to the data box, okay? Or to the promoter element, part of the promoter element. And then the A will come in, followed by the B to form a stable ternary complex. So there are three now. B, A, B. Okay. So it is the DAB ternary complex that will now attract and tether the pole 2, RNA polymerase 2, and the F complex to the promoter site. Okay. So the transcription factor 2F is sort of like the sigma factor in the prokaryotic because that is the one that is needed to recognize the promoter site. Then after that, the E will follow and then the H will follow. Or they can come together because the action of the E is to increase the kinase activity, but the actual kinase activity is the action of H. Okay. So which of this transcription factor is very important for transcription to happen? It's true that it needs to come together, but as they come together, the more important one, of course, is the F, which acts as the sigma factor, and then the H, which will put the phosphate group as the CN, so that RNA polymerase 2 can become active and bring about transcription of the hnRNA and mature to become the mRNA. Okay, but everything has to come together one at a time. Okay, what is post-transcriptional modification? Splicing of the exon. It will require yours to form your spliceosome. The spliceosome is consists of your primary transcript, the small nuclear RNA, and protein. So what will it do? It will actually remove or pick up the exons and bind it together. So in essence, it is removing the intervening sequence found in the primary transcript. Then terminal addition or modification can happen on the CAAN. This CAAN is particularly found in the tRNA, right? And this is where amino acid will be attached, the cognate amino acid. Then there are some nucleoside modification, as we pointed out in the discussion of the minor nucleotide found in the different nucleic acid. Then we have the precursor processing, the 45S RNA becoming the other 3 rRNA except 5S rRNA. Then higher conformation would be true for the tRNA. It is only in tRNA wherein we find double-stranded element, which is not found in the other RNA. Then formation of the RNA protein complex, this refers to the formation of the ribosome. The 4 RNA plus the protein complex, okay? Protein to form the ribosome. And then finally, we have the so-called RNA editing. Okay. So meaning to say, uh, splicing of the exons is very important. Coupled to, of course, you have to break the connection, remove it, and then connect. So what are the other processing that can happen? You learn that the mRNA is made up of the 3' poly A tail, and at the 5' end is the 5' capping of the 7-methyl guanosine tricoside. So this structural characteristic is peculiarly found only in mRNA. So you have to remove the introns. So the splicing is to remove the introns at the same time 5' capping and 3' poly A tail will happen. This is the editing, best exemplified by the formation of your B100 and B48. Okay. So the B100 is an apoprotein. It is found in the LDL. Okay. It is derived from the codon. Okay, when it is translated, you have this very long amino acid, 4,563 amino acids. Okay. So this will happen in the liver, the formation of your apoprotein B100. Okay. So in that long chains of codon, we have the CAA, which normally code for glutamine. In the intestine, RNA editing will happen. So what happened is that the C will become U, which is now UAA codon, which is a stop codon. So the apoprotein B that is formed is shorter, and it is known as your B48. Okay. So the function of the B48 is it is needed. This is found in the chylomicron for the absorption of the lipid from the intestine. So that is a very good example of your RNA editing. Never mind about the inhibitors. So the last topic is on genetic code. So this is the genetic code. It unmixed family and it mixed family. The unmixed is the serine, proline, threonine, alanine. Serine, there, 1, 2, 3, 4. So 16 boxes. So these are how many? 64 codons. Of the 64 codons, 3 are the stop codon. UGA, UAA, UAG. Okay. UAA, UAG, and then you just reverse UGA. So these are the 3 stop codons. So these are the punctuation codon is stop. Take note that the UGA per se, if it is in the mitochondria, it is a code for tryptophan. there is what we call the unusual tRNA, and UGA is the code that it will recognize, but there is something else in the mRNA that it can recognize for it to bring in your selenocysteine as an amino acid residue of the selenoprotein. Okay. We have how many bases? U, C, A, and G. Okay, so there are 4 bases. If you take one at a time, it will only give you 1. And we have how many amino acid? 20 amino acid. Take 2 at a time, it is not enough. We should take 3 at a time, make different combination of the 4 base to come up with the 64 codon. Then you have more than enough of the amino acid. First, it will tell us that a specific codon will code for a specific amino acid. If it is UUU, it is phenylalanine. If it is UUC, it is also phenylalanine. But if it becomes UUA, it is not phenylalanine anymore. So phenylalanine can be coded for by UUU or UUC. So it means to say it is the 3rd base that is not very stringent, okay? This is what we call as the degeneracy of the genetic code. When we say it is specific, then it is not ambiguous. Okay. So we have this one. The messenger RNA is read or translated by the ribosomal tRNA machine. The reading of the information is accomplished by the recognition of the genetic code. So first, it is universal. The nuclear DNA is the same in man as in other animals. It is read as a sequence of 3 bases known as the triplet codon in the mRNA, because the mRNA serve as the template. So 5' to 3' direction. Again, then read end to end in non-overlapping fashion. Important to remember that there is no comma. You mean to say you read 3 at a time in the mRNA. If one base is lost, you continue to read 3 at a time. So you can imagine the reading frame will now be different. And you will now be coding entirely for different amino acid and entirely for a different kind of protein. It's said to be degenerate but not ambiguous. Degenerate more than 1 codon for a given amino acid, not ambiguous, 1 particular codon specified for 1 and only 1 amino acid. The 3rd base is less specific. It means it will wobble. So wobble refers to the non-Watson-Crick base pairing that can take place on the 3rd position of the codon. This is for kinetic consideration, but more importantly, to minimize the deleterious effect of mutation. So meaning to say, if a mutation on the 3rd base will happen, and that particular amino acid that it is supposed to code comes from an unmixed box, then it means to say, okay lang, because it will code still for the same amino acid. Follow that? Let's say the U become a C, A, or a G. It is still serine. So there is a single mutation that happened on the 3rd base, but in the final end of the translation, it still code for the same serine. And that's the end. By the way, there is a table in replication in Harper. And what is the last step of replication? Let me see if I have it here. There is okay, it's not here anymore, but the last step in the table is telling you that there is now the association of to form it into how it should look like in the interface, okay, the chromatin formation. Any question? Dr. Pipito? Is Dr. Is Dr. Success around?
Yes, Doc.
Okay, so you go ahead. Gawas na ko ha. I will stop share.
Okay, Doc.
Okay, sige. Thank you. I will leave na.
Good morning.
Good morning,
Morning,
Doc.
Doc.
Good morning, Doc.
Morning, Doc.
Okay, so this morning we will talk about molecular genetics. So Dr. Meneses already discussed earlier about the DNA replication organization and everything, right? Hello?
Yes, Doc.
Okay. So basically, this lecture will just be a short one. We will talk about molecular genetics. So we'll just do some quick review on nucleic acids, your DNA or RNA, as well as the chromosomes. But this will not really take very long, as Dr. Menezes already um, mentioned more about DNA. Okay. So when we talk about molecular genetics, it is the study of the molecular structure of your DNA. It also includes its cellular activities, including its replication, and also the influence or its influence in determining the overall makeup of an organism. So how genetic information is transmitted and expressed on the molecular level is also a is also what molecular genetics is all about. So when we talk about nucleic acids, they are naturally occurring chemical compounds that serve as the primary information carrying molecules in the cells. So they typically play an especially important role in directing your protein synthesis. So when we talk about macromolecular structures, these are responsible for the storage as well as expression of all the genetic information that is necessary for building and maintaining life. So there are two main classes of your nucleic acids: your DNA, your deoxyribonucleic acid, which is considered as the repository of your genetic information, as well as the chemical basis of heredity, and your RNA, or your ribonucleic acid, which serves as vectors and translators of the information. And it also is responsible for the selective expression of the genetic information. So your DNA, it is when we talk about the chemical nature of the monomeric deoxynucleotide units of your DNA, they are your deoxyadenylate, your deoxyguanylate, no, all those um, your deoxycytinylate as well as your thymidylate. So they are held in polymeric form by the 3' 5' phosphodiester bonds, constituting a single strand. So they possess polarity, and the polymers, as depicted, possesses a polarity as mentioned, and one end has a 5' hydroxyl or phosphate terminus, while the other end has a 3' phosphate or hydroxyl terminus. So a segment of one strand of your DNA molecule, in which your purine and pyrimidine bases, your guanine, your cytosine, your thymine, and adenine are held together by this phosphodiester backbone between your 2' deoxyribosyl moieties and that are attached to the nucleobases of your N-glycosidic bonds here. So you take note that the phosphodiester backbone is negatively charged and also has a polarity. So this is a diagrammatic representation of your Watson and Crick model of your double helical structure of the B form of your DNA. So the two strands of these double-stranded helix are held register by both your hydrogen bonds between the purine and pyrimidine bases of the respective linear molecules and by your van der Waals forces and hydrophobic interactions between the stacked adjacent base pairs. So the pairings between your pyrimidine and purine nucleotides on the opposite strands are very specific and are dependent on hydrogen bonding of A and T as well as G and C. No? So these base pairings are often referred to as your Watson-Crick base pair, as what was mentioned by Dr. Meneses in her discussion. So examination of the model of your DNA reveals a major groove as well as a minor groove that winds along the molecule parallel to the phosphodiester backbones. So a single turn of your B-form DNA about the long axis of the molecule contains about 10 base pairs, and the distance that is spanned by one turn of your bDNA is 3.4 nanometers. And the width or the helical diameter of the double helix in bDNA is about 2 nanometers. Now, the classic Watson-Crick DNA base pairing between your complementary deoxynucleotides involves the formation of your hydrogen bond. So two such hydrogen bonds form between the adenine and thymine, and 3 hydrogen bonds are formed between your cytidine and your guanine. So in the double-stranded molecule, there is a restriction that is imposed by the rotation about your phosphodiester bond, the favored anti-configuration of the glycosidic bond, and the predominant tautomers of the 4 bases, your A, your G, your T, and C, that allows A to pair only with T and G only with C. So this is basically very specific pairing. Now, the human mitochondria contains about 2 to 10 copies of a small circular dsDNA molecule that makes up approximately 1% of your total cellular DNA. So this codes for mitochondrial specific ribosomal and transfer RNAs and for proteins that play key roles in the respiratory chain. So these are the features of your human mitochondrial DNA. So take note of these different features of your mitochondrial DNA. So I won't go over them individually. On the other hand, your RNA is a polymer of purine and pyrimidine ribonucleotides that is linked together by a 3' 5' phosphodiester bonds. And this RNA is analogous to those in the DNA. Now, how does it differ from your DNA? So although the sharing of many features, or though there are features that are similar between your DNA and RNA also possesses several specific differences. So in RNA, the sugar moiety is ribose. It contains the ribonucleotide of uracil. It exists as a single strand. Its guanine content does not necessarily equal its cytosine content, nor does its adenine content necessarily equal its uracil content. It can also be hydrolyzed by alkali to 2' 3' cyclic diesters of your mononucleotides. Although the RNA typically exists as a single strand, given the proper complementary base sequence with opposite polarity, the single strand of this RNA is capable of folding back on itself. So it can acquire a double-stranded characteristics with G pairing with C and A pairing with U. So basically, this diagrammatic representation of the secondary structure of a single-stranded RNA molecule shows a stem loop or the hairpin like structure that is formed, giving the double-stranded characteristics of your RNA. So this is basically what causes your RNA to acquire its double-stranded characteristic. So the formation of this structure is basically dependent on the indicated intramolecular base pairing that is seen in this diagram. No, there are base pairing that occurs in your hairpin-like structure. So there are different classes of RNA in all prokaryotic and eukaryotic organisms. No, these are your mRNA or your messenger RNA, your transfer RNA or tRNA, your ribosomal RNA, and your small RNA. So each differs from the others by its abundance, the size, the function, as well as the general stability. So first, let's talk about your messenger RNA. So this is the most heterogeneous class in terms of abundance, size, and stability. So this functions as messengers conveying the information in a gene to the protein synthesizing machinery, where each mRNA serves as a template on which a specific sequence of amino acids is polymerized to form a specific protein molecule, which is your ultimate gene product. So these mRNA represents 2 to 5% of the total eukaryotic cellular cellular RNAs. So the 5 terminus of your messenger RNA is capped by a 7-methylguanosine triphosphate that is linked to an adjacent 2' O-methyl ribonucleoside at its 5' hydroxyl through the 3 phosphatases. So the cap is involved in the recognition of your mRNA by the translation machinery, and also helps to stabilize the mRNA by preventing the nucleolytic attack of your 5' exoribonucleases. So the protein synthesizing machinery begins translating the mRNA into specific proteins beginning downstream of the 5 prime or cap terminus. So the poly A tail at the 3 prime end of mRNAs maintains the intracellular stability of the specific mRNA. This is done by preventing the attack of the 3 prime exoribonucleases and will also then facilitates the translation. So both the mRNA cap and the poly A tail are added post-transcriptionally by the non-template directed enzymes to mRNA precursor molecules. Now let's talk about the transfer RNA. So the transfer RNA serves as adapters for the translation of the information in the sequence of nucleotides of the mRNA into a specific amino acid. So this vary in length from 74 to 95 nucleotides and is composed roughly of 20% of total cellular RNA. So the primary structure, that is the nucleotide sequence of all tRNA molecules, allows extensive folding and intra-strand complementarity to be able to generate a secondary structure that appears in 2 dimensions like a clover leaf. And this contains 4 main double-stranded arms or stems. So all your tRNA molecules, as mentioned, contains 4 main double-stranded arms or stems, and they are connected by a single-stranded loops named for their respective nucleotide composition or function. So you have your acceptor arm, your T-psi-C arm, or your translation arm, your D arm as and your anticodon arm. So your acceptor arm is the site of attachment of the specific amino acid. Your translation arm is responsible for the binding of your amino acyl tRNA to the ribosomal surface. Your D arm is responsible for proper recognition of a given tRNA to its amino acid tRNA synthetase, and your anticodon arm recognizes the triplet codon on your mRNA. Now, your ribosomal RNA is a class of RNAs that are being assembled together with your numerous ribosomal proteins to form the ribosomes. So we all know that a ribosome is a cytoplasmic nucleoprotein structure that acts as the machinery for the synthesis of your proteins from mRNA templates. So the rRNAs represent roughly 70% of the total cellular RNA, and this is highly methylated and is packaged in the nucleolus with the specific ribosomal proteins. Now, your small RNAs are complexed with proteins to form your ribonucleoproteins and are distributed in the nucleus, the cytoplasm, or both. So they range in size from 20 to 1,000 nucleotides and is present in 100,000 to 1 million copies per cell, collectively representing less than 5% of cellular RNAs. So your small nuclear RNAs is significantly involved in rRNA and mRNA processing and gene regulation. So these are some of the specific, rather species of small stable RNAs found in the mammalian cell. So you have your U1, U2 until U6. You have your 4.5S and 7SK. So your U1, U2, U4, and your U5 and U6 are involved in intron removal. So U1, U2, U4, U5, and U6, they are responsible or involved in intron removal and the processing of mRNA precursor into mRNA within the nucleus. Your U7 snRNA is involved in the production of the correct 3' ends of histone mRNA, which lacks a poly A tail, and your U7sk RNA associates with several proteins to form a ribonuclear protein complex that modulates the mRNA gene transcription elongation by RNA polymerase II. So take note of these different snRNA species and their localization. Now you also have these non-coding regulatory RNAs. Um, They are also called or divided into your microRNAs and silencing RNAs. So their role basically is to inhibit gene expression at the level of specific protein production by targeting mRNAs, and they are hybridized via the formation of RNA to RNA hybridization to their targeted mRNAs. Now, you also have your large ncRNAs, which includes your circular RNAs and long non-coding RNAs. These, on the other hand, play an important role in cellular biology by regulating gene expression. So specifically, your circular RNAs produced by the RNA splicing type reactions from a wide range of precursor RNAs. So they are particularly abundant in cells of the nervous system. Your long non-coding RNAs do not code for protein. They only contribute to the structural aspects of the chromatin, as well as the regulation of your mRNA gene transcription by your RNA polymerase II. Now let's talk about the chromatin. So chromatin are chromosomal materials in the nuclei of cells of eukaryotic organisms. They consist of very long double-stranded DNA and a nearly equal mass of small basic proteins termed histones, as well as a smaller amount of non-histone proteins and a small quantity of RNA. So in chromatin, core particles are separated by a roughly 30 base pair regions of DNA that is termed as a linker. So your histones are small family of closely related basic proteins that is the most abundant chromatin proteins, and its organizational unit of this soluble chromatin is the nucleosome. The nucleosomes are composed of DNA wound around an octameric complex of histone molecules. So they are approximately 10 nanometer in diameter and are connected by the DNA filaments. They contain four major types of histones, your H2A, H2B, or H3, and your H4, as they are often called as the core histones. So your core histones are subject to at least 6 types of covalent modification or post-translational modifications, PTMs. And these are the processes that they may be involved: acetylation, methylation, phosphorylation, your ADP ribosylation, monoubiquitination, and sumylation. So this table shows to us the possible roles of your post-translationally modified histones. So when you talk about acetylation, as I mentioned earlier, the acetylation of your histones H3 and H4 is associated with the activation or inactivation of gene transcription. The acetylation as well of your core histones is associated with chromosomal assembly during your DNA replication. Another process is the phosphorylation. No, your phosphorylation of your histone 1 is associated with the condensation of chromosome during the replication cycle. There is also ADP ribosylation of histones, which is associated with your DNA repair. Your methylation of histones is correlated with the activation and repression of your gene transcription. The monoubiquitination is associated with gene activation, repression, and heterochromatic gene silencing. And the sumylation of histones is associated with transcription repression. So basically, there are different roles of these post-transitionally modified histones when they undergo specific process, no, or metabolism. So now let's talk about the higher order structures, which provide for the compaction of your chromatin. Now, each phase of condensation or compaction and organization decrease the overall DNA accessibility to an extent that the DNA sequences in the metaphase chromosomes are likely almost totally transcriptionally inert. So here, the chromosomal DNA is packaged and organized at several levels, and each phase of the condensation, as I mentioned earlier, or compaction of organization will decrease the overall DNA accessibility. Now, this table shows to us the packaging or compaction ratios of each of the orders of DNA structure. So take note of these different chromatin forms and the responsible packing ratio of these chromatin forms. Now, your DNA is organized also into chromosomes. So these chromosomes possess a twofold symmetry with the identical duplicated sister chromatids. And each sister chromatid contains one dsDNA molecule. So here, the centromere, the chromatids are connected at a centromere, no? Here, the ends of each chromosome contains the structure also called as the telomere. So the telomeres consist of short PG-rich repeats, while your centromeres are adenine thymine-rich region containing the repeated DNA sequences. So basically, that's basically the things that you need to know, and in general, the things that you need to know about molecular genetics. So I guess that ends my correlate for this morning. Will there be another correlate after me?
No questions for Dr. Salces. Dr. Meneses is not here anymore, no?
Ah, yes, she left already. So will there still be another correlate after me, Gwen?
Wala na niya. But
Okay.
do you like, okay, if you don't have any questions for Dr. Salces, you like to review on this morning's exam? Okay, I'll start with the review. Thank you, Dr. Salces. I think they don't have any questions. We're not overwhelmed lang sa exam this morning. The exam this morning is very difficult.
Yes, Doc.
Yes, Doc.
Yes, Doc.
Yes, Doc.
What
Yes,
is
Doc.
difficult in the exam? All of it?
Yes, Doc.
Everything.
Okay, sige, we'll go through it one by one. For my topic, I can only answer your queries, but I can relate to Dr. Borromeo. Actually, Dr. Borromeo sent me a rationale for her exam. So if you have questions on specific items. Okay. So which of the following can help you determine the shape of a protein? Answer to this one is to determine the specific sequence of amino acids in protein. What are the choices? Other choices: through hydrolysis to find out if the protein contains other elements aside from amino acids, to determine the Ramachandran plot of the protein, and to predict if motifs are present in protein or not. So answer is to determine the specific sequence of amino acids in protein. Any question or concern to the item? Okay. Number 2. Which of the following separatory or purification techniques of proteins is specifically based on their Stokes radii? Answer: ultracentrifugation, SDS page, HPLC, gel permeation chromatography. What's your answer?
Gel filtration, Doc. Gel filtration chromatography. Doc.
Okay. I have to confirm with Dr. Meneses. She did not give, to be honest, she did not give a key to this one. And then she's not here. But I placed the correct choice item with gel chromatography. But this has to be checked with Dr. Meneses. Okay. Next item, a single polypeptide chain protein must assume which structure to become functional? So this, I think, is an easy question. What is your answer?
Tertiary.
Tertiary structure. So that's the correct answer. Which of the following participates in the correct intra and enter disulfide bonds present in an Ig or immunoglobulin molecule? What is your answer? Anyone? Is it disulfide isomerase, glutathione, both, or neither? I think this is disulfide isomerase. What's your answer? This is a difficult one. So the answer, correct answer, is both disulfide isomerase and glutathione. Okay. 5. What is the usefulness of the Ramachandran plot in understanding the different conformational structures of proteins? So the answer is to determine the predominant secondary structure of a protein. Questions so far? Okay, next. Why must acid-base balance of the body be maintained at physiologic pH? Answer: the shape and function of particular proteins are governed by the pH of the environment. Question so far? None. Next. The two-dimensional electrophoresis is used in the separation of complex mixtures of proteins wherein the correct answer is first dimension is isoelectric focusing and second dimension is SDS page. Okay, next question. Which of the following is the most important bonds needed for the tertiary structure of protein to assume the globular shape? Answer: hydrophobic interactions or effect of alkyl R groups of AA amino acid residues. 9. Which of the following depicts the specific molecular abnormality found in prion diseases? What's your answer? Actually, Dr. Meneses did not give me a specific choice for this, the answer key, but I place incorrect. The so-called PRP-SC acts like an infectious agent and transforms all other PRPC. So if there are changes, then it will be automatically, I'll correct. Okay. Number 10. Yes. So in protein folding, to ensure the formation of globular protein, which of the auxiliary protein and enzymes involved in protein folding must act sequentially? Answer: HSP70, then chaperonin. Next item. Which of the following bonds present in nucleoside triphosphate has a high phosphoryl transfer potential? Answer: acid anhydride bond between beta phosphate and gamma phosphate. Next question. The TMP found in tRNA molecule is due to the, what's your answer? The answer is post-transcriptional addition of methyl group from SAM as a substituent group of C5 of uracil in UMP. Next item. CDP derivatives is used in the synthesis of which of the following? Answer: phospholipids. Correct? But this is a difficult question for you based on the result of the exam, but this is part of the synthesis also of the phospholipids, right? Which of the following two properties in the structures of the monomeric nucleoside monophosphates ensure the hydrogen bonding between complementary bases found in the double-stranded DNA? Answer: predominant tautomers of keto and amino in the base and anti-beta and glycosidic band configuration. Which enzyme abnormality is present in a boy with gout, mental retardation, and peculiar behavior of self-mutilation? Answer: complete absence of HGPR taste. Which of the following statements is not true for both purine and pyrimidine anti-nucleotide de novo biosynthetic pathways? Answer: both require THF to carry one C group to form major purine and pyrimidine NT. Which of the following synthetic nucleotide analogs mechanism of action can least affect the NT metabolism? Answer: methotrexate is to inhibits thioredoxin reductase. The question is, can least affect? 18. Which of the following least likely depicts the source of the substituent group found on the purine and pyrimidine-based NTs? Answer: the amino substituent of uracil is derived from ASN. Next item, which of the following best describes the difference between the purine and pyrimidine NT de novo biosynthetic pathway? Answer: the role of THF participation and as a 1C group carrier in purine NT and as a source of methyl group to form TNP. What is the medical reason why a physician asks the patient with gout to limit his/her intake of alcoholic drinks? Answer: metabolism of alcohol by the liver will result in lactic acid formation, leading to renal hyperuricemia. Next question, which of the following best describes the formation of the deoxyribonucleotides? Answer: two reductants are needed, the immediate reduced thioredoxin and the ultimate non-pH. PRPP GLN amido transferase is best described in which statement? So the answer is both A and B, which it is a rate-limiting enzyme of the purine and T de novo biosynthetic pathway. It is the catalyst that allows the putting in of N9 from amide group of GLN on the purine ring. Which of the following abnormalities in PRPP synthetase leads to metabolic hyperuricemia? Answer: neither A and B. So neither the decreased Vmax of the enzyme and increased Km of the enzyme. So you have a figure, you had a figure. So that figure is which, okay, I'll try to show how the figure, if I can open it. So this is the figure. And the question is, so under which EC classification will the enzyme catalyzing the reaction below belong to? And the answer is EC5. And then you have another figure. Okay, so that figure. And then, so the carboxypeptidase A catalyzes the hydrolysis of peptide A, B, C, D in a 10 mm phosphate buffer. Under the condition of the experiment, the following data points were recorded. Thus, your figures, the values. What is the Km for the carboxypeptidase for peptide A, B, C, D? So the answer is 1 times 10 to the negative 5 million. 26, an antibiotic being developed in a closed transition state analog to the substrate of the enzyme that participates in cell wall synthesis. Binding of the antibiotic can reduce enzyme activity without changing the enzyme structure. Also, the activity is restored when more substrates are added. Which of the following statements is true for the antibiotic described? Answer: it increases the apparent Km of the enzyme. So what is your, any questions? So I'll read the rationale of Dr. Borromeo for that question, and under which EC classification would the enzyme catalyzing the reaction below belong to? So the answer is EC5. The enzyme is an isomerase, so that's an EC5. And then so regarding the carboxypeptidase A that catalyzes the hydrolysis of peptide A, B, C, D. So the answer is 1 times 10 to negative 5. So assume that Vmax is 140 micromole per liter. Half the maximal velocity is 70. Km is the substrate concentration at half Vmax. So the answer is 1 times 10 to the negative 5. So regarding the antibiotic being developed is a closed transition analog to the substrate. So the question is, which of the following statements is true for the antibiotic described? So the answer is it increases the apparent Km of the enzyme. So rationally, this is a competitive inhibitor. So as she mentioned during the lecture, that competitive inhibitors can be transition state analog. So naturally, they bind at the active site. Like competitive inhibitors, they do not change or induce any, or do not induce any conformational change, but activity is reduced due to reduced substrate binding. The fact that activity is restored when more substrates are added means that the analog is not covalently bound to the inhibitor and characteristic of competitive inhibition. Any questions so far? I'll proceed. So a reaction catalyzed by enzyme A has a Km value of 2 times 10 to negative 6 and Vmax of 30 millimole. At a concentration of 2 times 10 to negative 3, the rate of the reaction will be, answer to this is 30 millimole. So rationally, so you didn't have to solve this. The fact that that something is raised by 10 to the negative 3 versus 10 to the negative 6 tells you there is a huge increase in S, or the substrate, and the enzyme is most likely operating at its Vmax, which is 30 millimoles per liter. Next question, how does optimal temperatures for a reaction change with and without an enzyme catalyst? Answer is the optimal temperature is lower with a catalyst than without. So rationally, uncatalyzed reactions create the rate with increased temperature. There is no clear limit. but enzyme catalyzed reactions usually operate at lower temperature due to risk of denaturation. Which of the following is not a method by which enzymes can decrease the activation energy of reactions? Answer: breaking chemical bonds in the enzyme to provide energy. So rationally, enzymes decrease the activation energy by 4 modes of catalysis. Acting as electron donors and acceptors as choice, it is a definition of acid-base catalysis. And then forming transient covalent bonds with the substrate, this is covalent catalysis. And then positioning of substrate in the right orientation, it is by proximity and orientation. And then the correct answer, breaking chemical bonds in the enzyme to provide strain catalysis, is breaking of substrate bonds, not enzyme bonds. The bonds within the enzyme within the enzymes are binding. So the reaction where acetyl-CoA is converted to malonyl-CoA in fatty acid synthesis has a GO of positive 385 calories per mole. For the reaction, what is the most likely KAQ for the reaction? The answer is 0.5. So, rational C relationship of standard free energy and KAQ. If the standard free energy is negative, the KAQ is more than 1 and favors product formation. If the standard free energy is positive, the KAQ is -1 and favor substrate or none in non-spontaneous reaction. So next question, when plotting and solving the Lineweaver-Burke plot, which of the following statements is true? Answer: Km equals slope times 1 over y minus intercept. So choice, the slope represents the Hill equation, that is slope is Km over V max. And then choice that the y-axis is the velocity of the reaction, that's y-axis is 1 over V. Choice where the x-intercept is the Km, so that's -1 over Km. And then the answer, where is Km equals slope times the y over the reciprocal y-intercept. You can derive the equation if you have the value of the slope and V max. So slope is what is equal to Km over V max. So Km is equal to slope times the V max. The y-intercept represents the value of 1 over V max. But in the Lineweaver-Burke plot, the values are reciprocal. So Km is equal to slope times y over the y-intercept. So next question, which formation of regulation will take the longest time? Answer: induction of transcription. Rationale, all are quicker except for induction and repression of transcription. Next question, Daniel Koshland's model of enzyme substrate binding refers to the fact, answer: substrate binding promotes conformational change in enzymes and optimal orientation of catalytic groups. So rationale, Daniel Koshland's model is the induced fit model. So the induced fit model is likened to a glove on stacking, where the enzyme changes or adjusts its shape to mold into the substrate. So the choice where enzyme substrate binding increases the reaction entropy or randomness. So enzymes lower activation energy, therefore they decrease entropy or randomness of the transition state. And then choice stereospecificity is observed during enzyme substrate binding. So stereospecificity is reflected more by the lock and key model. And then on the choice, enzyme substrate binding induces movement along the reaction coordinate to the transition state. So that's the general action of the enzymes. Questions or clarifications before I proceed to carbohydrates and lipids. Okay, none. I'll proceed. Which reducing sugar is a disaccharide? Answer is maltose. Which of the following molecules is not a derived monosaccharide? Answer, lactulose. Which statement best describes physiologically important monosaccharides? Answer, D-mannose, D-xylose, and D-arabinose are constituents of glycoproteins. Which of the following statements best describes glycosidic bonds? All sugar-to-sugar glycosidic bonds are O-type linkages. Which statement best describes sugar alcohols? Answer, examples of sugar alcohols are sorbitol, mannitol, and xylitol. Which statement best describes biochemically important sugars? D-ribose is present in nucleic acids, ATP, and flavin coenzymes. Which of the following statements best characterizes the chemistry of carbohydrates? Answer, galactose, fructose, glucose, and mannose are all isomers of each other. Questions, clarifications? Okay, which of the following lipids, which of the following best describes the configuration of the serine nucleus found in their body? Answer to this is the junction of the A and B rings is cis. So it can be in the cis or in the trans. And the rings are in the chair form, right, to make it stable. So beta bonds are shown by hard or bold lines. So which of the following is true of polyprenoids? So they have the same parent compound as cholesterol, but they are not considered steroids. Which of the following antioxidant is a food additive? That's propyl gallate. What is the major lipid in fat deposits? Answer, triacylglycerol. Which molecule is common precursor to both triacylglycerol and phospholipids? That's glycerol 3-phosphate. Which of the following is true of glycolipids? Answer, GM3 consists of ceramide and one molecule each of glucose, galactose, and sialic acid. So nomenclature of that fatty acid that was shown, that is 20 carbon, then 4, that's all cis. 4 double bonds cis, it starts at 5, 8, 11, and 14. Which of the following is true of polyunsaturated fatty acid? So they are precursors of the thromboxanes. So a 56-year-old male consults you for his high cholesterol, hypertensive, and then has occasional chest pain. So the ideal from the choices is mackerel oil. Last number, which of the following phospholipase degrades lysolecithin? So your lysophospholipase is actually as phospholipase B. When you see it in your book, diba, it's a phospholipase B. It claims the SN1 of your phospholipid carbo SN1. Any question?
Hello, Doc.
Swin, yes.
Good morning, Doc. Can we go back to the question where the answer was fructose, galactose are isomerizes, Doc?
Mhm. That is the answer, Jasmine, no?
Yes, Doc, but I would like to clarify lang, Doc, ba?
Rita. What other choice can you remember?
I think I answered galactose is an epimer of glucose, Doc.
Galactose.
At carbon 4.
Rita. Which of the following statements best characterizes the chemistry of carbohydrates? So the key here is galactose, fructose, glucose, and mannose are all isomers of each other. So this, for you, this is a wrong statement.
I think, Doc, that's also correct, but I think po na ang katong galactose is an epimer of glucose at carbon 4. If wala ko masayo.
That is other choices. Glucose and galactose are C4 enantiomers
Ay, okay,
of
sorry.
each other.
Yes, Doc, okay na day. Enantiomers day ang word. Sorry, ako ang sayo.
Sige, sige.
Thank you, Doc.
Okay, any other clarification for a particular item or choice? None? So that we can end and you can rest for a little and then study tonight again for tomorrow's activity, PBLB. Any other concerns? For long exam 1, there's one bonus. The one that Harvey questioned on, that one when you put pressure on a solute, you cannot remember the specific, the one with the figure but on cellular activity, that Harvey answered stop, and it's the correct key that was given. I asked Dr. So it was supposed to be the correct answer for that is reverse. So it will be given a bonus and your scores will be adjusted. Okay. Any question for this exam? Okay, my suggestion is review on your mistakes and why you have that particular item wrong. Don't dwell on your scores too much, but see on what you can improve on the next succeeding exam. Okay, if you're down for today's score, be down for 1 to 2 hours, have a rest. But my suggestion is you have to get up and then start over again and do your best on the succeeding exams. Okay? Namiyaw sila.
Hello, Doc.
Yes, this is Queenie.
Yes, Doc. May I
Okay,
just ask, Doc, if there will be an extension for the deadline for the CMAP so that we could focus on studying in the afternoon of the PBL?
Queenie,
Yeah,
for that?
I would like to ask, Doc, if there is an extension for the deadline for the CMAP, the concept map, Doc,
Concept map for when?
for tomorrow's PBL, Doc.
You can do it after the unit exam. Submit it after the unit exam.
Okay, Doc. Thank you, Doc.
Okay. Sige, any concerns? Reminder. When you have difficulty logging in or opening in the app for the exam, please do not message us anymore sa email. Please join us here in Zoom. Okay, so that we can answer your queries and help you troubleshoot and go in sa exam. Okay,
Doc, I just want to ask, Doc,
James.
for the e-portfolio, it will be today the deadline, Doc.
You can extend it. Who asked? James, who's your mentor?
Doc Lasaka.
Doc Lasaka, you can extend it after the unit exam.
Okay, thank you, Doc.
Anything else? None? If none, then you can go and have a rest. Take your lunch and have a rest. Okay, bye everyone.
Thank you, Doc.
Thank you, Doc.